One Tuesday afternoon, Briggan sits and thinks about how attractive she is.
Briggan, I am way more attractive than you are.
Please.
Really? Have you had a look at this? This is just amazing.
You're kidding, right?
No. I can explain you with just genes and genes expression why I am just so amazing. Hey, how's it going? I'm Dr. Brent Poss, and this is an educational video on gene expressions. Because trust me, I'm a doctor.
Gene expression in its simplest form is found in prokaryotes, even though that's still pretty complicated. Prokaryotes use gene expression to change their metabolic rate in accordance to the environment. Although, if they didn't, they'd fall over and die. Which is obviously a figure of speech, because prokaryotes really can't fall that far.
Many genes are turned on and off due to the environment. The entire stretch of DNA that contains the operator, the promoter, and the subsequent DNA is called the operon. The switch to turn on and off the operon is the operator, and the promoter is the site that starts transcription.
A repressor is the protein that controls whether the operator is on or off. If the repressor is bound to the operator, it blocks the attachment of RNA polymerase to the promoter. Repressors are very specific and only bind to certain operators.
Repressor proteins are created by a regulatory gene. The repressors are created at a constant rate and used when needed.
Repressor proteins are helped by corepressors, which are small molecules that cooperate with repressors to alter the operon.
In special situations such as the lac operon discussed later, a special small molecule, called an inducer, inactivates the repressor.
Bacteria are dependent on nutrients such as the amino acid tryptophan. Without it, it can activate a metabolic pathway to create its own tryptophan. Surrounded by it, the metabolic pathway is shut down because it's not needed to save the wasting resources. When tryptophan is absent, the repressor isn't active. This makes the operon turn on, producing the amino acid. RNA polymerase attaches to the DNA at the promoter and transcribes the operon's genes. But when there is tryptophan present, repressor becomes active and the operon is off. As tryptophan accumulates, it inhibits its own production by activating the repressor protein. The TRP operon, or tryptophan operon, is described as a repressible operon. Transcription is inhibited when a specific molecule binds allosterically to a regulatory protein. Allosterically means that it changes the activity of a protein. The lactose operon is different than the tryptophan operon because the lac operon is described as an inducible operon, which is stimulated by a small molecule interacting with a regulatory protein. 3 enzymes take up and metabolize the lactose, and the genes for these enzymes are clustered in the lac operon. One of the enzymes is the LAC-Z, which codes for beta-galactosidase, which hydrolyzes lactose to glucose and galactose. The next is the LAC-Y enzyme, which codes for permease, which is the membrane protein that transports lactose into the cell. Finally, the LAC-A enzyme codes for an enzyme called transacetylase, whose function in lactose metabolism has not really been discovered. The gene for the lac repressor, LAC-L, is adjacent to the lac operon. When lactose is absent, the repressor protein is active and the operon off. The lac repressor is innately active, and in the absence of lactose, it switches off the operon by binding to the operator. But when lactose is present, the repressor protein is inactive and the operon is off. Or is on. Lactosidase, an enantiomer of lactose, de-represses the operon by inactivating the repressor. In this way, enzymes for lactose metabolism are started up.
Eukaryotes are much more complex than bacteria, so naturally gene expression in them is too. The chromatin structure is the successive layers of DNA packing. Histones are positively charged amino acids which are bound together in 4s to the negatively charged double helix DNA, which creates a bead called a nucleosome.
The 5th histone, histone H1, is an extra little one that binds to the DNA adjacent to the bead. And then the 30 nanometer chromatin fiber is a bunch of histones coiled together.
Loop domains are basically these 30 nanometer fibers bound in a scaffold shape with non-histone proteins.
The final phase of the histone is the metaphase chromosome, which is the X shape that you get.
Gene expression must be controlled on a long-term basis in order to create cellular differentiation, which is the divergence in form and function for the specialization of cells. Gene expression is controlled in a number of ways.
DNA methylation takes a methyl group and adds it onto the end of the bases, which makes sure the genes are not expressed.
Histone acetylation is when an acetyl group is added to the histones to loosen their grip to allow transcription to occur more easily. When the acetyl group is taken off, then the grip is tightened of the histones and it represses transcription.
Control elements are segments of non-coding DNA that help transcription by binding with transcription factors.
There are two types of control elements: proximal, which are close to the promoter, and distal, which are also called enhancers. Enhancers help recognize transcription factors of DNA, and they stimulate the transcription of certain genes.
Activators help enhancers by telling them when to start transcription.
Gene expression is not only controlled by transcription. Regulatory measures can be used to essentially spell check the gene expression.
An example of gene regulation at the RNA level is alternative RNA splicing, in which different mRNA molecules are produced from the same primary transcript.
mRNA degradation is used to change the patterns of protein synthesis. Degradation of mRNA occurs more slowly in eukaryotes than in prokaryotes.
Transcription controls block the different stages of protein synthesis.
Protein degradation essentially checks to make sure that there's no proteins that are mismatched or broken. Those that are flagged and then eaten by proteasomes, and those that work are given a certain lifespan.
This has been an educational video from Dr. Brent Poss. Stay classy, America. See, Bergen, that video explained how I'm way sexier than you are.
Guess you're right. I mean, you can't really argue with that really attractive Dr. Brent Poss.
He's a good guy, a really good character, and I'm just straight up sexy.
Are you recording?
Yeah. See, Bergen, from that video, I'm just way.
A curve joke.